In this lecture, we will examine the gross and microscopic structure of skeletal muscle, levels of organization, and how each level functions to contribute to muscle contraction. There are four major functions of skeletal muscle: production of movement, maintenance of posture, stabilization of joints, and generation of heat for the body. In addition, muscle also serves as a nutrient reserve for protein and glycogen. Most skeletal muscles cross at least one joint. When the muscle contracts, it pulls on the bone, creating movement. Some skeletal muscles insert on skin, like the ones that cause changes in our facial expressions. Muscles maintain our posture against the force of gravity. Different parts of certain muscles are alternately contracting and relaxing, thereby making continuous adjustments that maintain our posture. Skeletal muscles are very important for stabilizing joints. Take, for example, the shoulder joint. As you learned in the last unit, the pectoral girdle and upper limb is well adapted for tremendous mobility, but this comes at the cost of stability. It is the muscles surround the shoulder joint rotator cuff that provide most of the stability for the joint, as well as creating movement of the upper limb. Muscles generate heat when they contract. We all have experienced this when we walk vigorously, jog, climb stairs, lift weights, etc. In fact, almost 75% of the ATP used to perform muscle contraction is lost as heat. Shivering when you are cold results from tiny muscle contractions that don't produce effective movement but serve to warm the body. Muscle is almost all protein, which can be used to make ATP under conditions of starvation or fad dieting, where a person loses weight too fast due to extreme calorie restriction. Glycogen is a storage form of glucose which can be mobilized to support ATP production, mainly to power muscle contraction. Muscle also can help to protect the internal viscera, and just think of the ventral surface of the abdominopelvic cavity. No bones there. In order for muscle to carry out these special functions for the body, it has some unique properties. First of all, it has the ability to shorten. We call this muscle contraction. It is excitable or irritable, which means that it has the ability to receive and respond to a stimulus like a neurotransmitter or pH change. It responds to stimuli by generating an electrical impulse or action. Potential nerve tissue is the master of this. Muscle tissue is extensible, which means that it can be stretched. We know that muscle shortens when it contracts, but it can be stretched beyond its resting length. To be able to create movement across a joint, muscles are arranged in opposing pairs on opposite sides of the joint. This means that when flexors contract, the extensors on the opposite side must relax or stretch so the movement can be made. Lastly, muscle is elastic. It has the ability to recoil like a rubber band and resume its resting length after it has been stretched. Although we normally don't think of muscle this way, each muscle is an individual organ of the muscular system and is made up of several kinds of tissues in addition to muscle tissue: blood vessels, nerve fibers, connective tissue. Each individual muscle is surrounded by a connective tissue sheath called the epimysium and consists of hundreds to thousands of individual muscle cells that are arranged in bundles called fascicles. Likewise, each fascicle is surrounded by CT called the perimysium. Each fascicle, which means stick, by the way, consists of numerous muscle cells. Muscle cells are long and cylindrical and are referred to as muscle fibers. But they are true cells with nuclei and other organelles, not extracellular fibers like collagen and elastin. The muscle fibers are surrounded by thin CT called endomysium, which also contains a stem cell called a satellite cell. Each muscle fiber is made up of rod-like contractile elements called myofibrils. These in turn are composed of sarcomeres, which are the structural/functional unit of skeletal muscle. The sarcomeres are made up of myofilaments of two types, thick and thin. The thick myofilaments consists mainly of myosin, whereas the thin myofilaments consist mainly of actin. The connective tissue forming the sheaths are important because they reinforce the muscle, preventing them from bursting during strong muscle contractions, and support each individual muscle cell or muscle fiber. They are also important because it is through the CT sheaths that blood vessels and nerves travel. Although we assign different names to the CT sheaths, they are all continuous with each other and divide the muscle down into smaller and smaller units, as discussed previously, for more efficient function, control, and blood supply. The outermost epimysium blends with the deep fascia that lies between neighboring muscles, or the superficial fascia deep to the skin. It also extends beyond the muscle to form tendons, rope-like arrangements of dense regular CT, or aponeuroses, flat sheets of CT. Tendons attach muscle to bone. They are smaller in size compared to bulky muscles, so more of them can pass over a joint. This is especially true of the muscles that control our wrists and fingers. The muscles are in the forearm. The tendons insert on the wrist, hand, or fingers. The next couple of slides are taken from your textbook and summarize the structure and organizational levels of skeletal muscle. It would be a good idea to learn muscle organizational hierarchy as soon as possible to avoid confusion later on. Many of the terms sound alike. The structural functional unit of striated muscle, be it skeletal muscle or cardiac muscle, is the sarcomere. Notice that each sarcomere is composed of myofilaments, which are the contractile proteins. Let's look now at the structure of an individual skeletal muscle cell or fiber. Each muscle cell is a long, multinucleated cylinder ranging from about 10 to 100 micrometers in diameter to as much as 30 centimeters 12 inches in length. Each muscle cell is formed during embryological development by the fusion of precursor cells called myoblasts. Under the light microscope, alternating light and dark bands are visible, which give skeletal and cardiac muscle a striated appearance. The nuclei are located peripherally just under the plasma membrane of the muscle cell. Why do muscle cells have so many nuclei? And why are they so long and skinny? The answers to these questions have to do with how skeletal muscle forms during development. As mentioned previously, each muscle fiber is formed from the fusion of embryonic mesodermal cells called myoblasts. In this diagram, the reddish myoblasts will fuse together, forming the mature muscle cell. If a muscle cell was made from 100 myoblasts during development, then this cell will have 100 nuclei. Note that there is one myoblast that is a tannish color. This cell stays in the embryonic state, meaning it does not fuse with the others, nor does it differentiate, and becomes a satellite cell. It is important that mature skeletal muscle fibers have many nuclei to provide multiple copies of the genes for contractile proteins. When one lifts weights, muscle cells get bigger because they synthesize more contractile proteins to meet the demand of the increased stress put on them. The satellite cells are stem cells that can become active under certain circumstances. For example, if the muscle fiber becomes damaged, the satellite cells can help to replace the injured tissue and repair the muscle cell. Muscle cells or fibers are so structurally and functionally specialized. There is a special set of terminology that is applied to skeletal muscle. So in muscle, the plasma membrane or plasmalemma is called the sarcolemma. Sarcos means flesh. Flesh is meat, and meat is muscle. Transverse or T-tubules are a unique modification of the sarcolemma. The cytoplasm is called the sarcoplasm. It is similar in composition to that of other cells, but it contains granules of stored glycogen and myoglobin. Myoglobin is a reddish pigment that stores oxygen similar to hemoglobin of RBCs. The sarcoplasm contains the myofibrils and a very specialized version of smooth endoplasmic reticulum called the sarcoplasmic reticulum. Let's look at these unique features of skeletal muscle fibers that are necessary for these cells to perform their specialized functions in the body. We will begin with the sarcoplasm. As we learned previously when studying the organization of muscle tissue, Muscle fibers contain numerous, densely packed cylindrical rods called myofibrils, each about 1 to 2 micrometers in diameter, accounting for about 80% of the volume of the cell. There are so many myofibrils that all other organelles, such as mitochondria, are crammed between them, and the nuclei are pushed to the periphery under the sarcolemma, as previously discussed. The myofibrils contain the myofilaments, which are the contractile elements of the cells. The myofilaments are organized into repeating units called sarcomeres. The sarcomere is the structural/functional unit of striated muscle. Skeletal and cardiac muscle are striated. Each myofibril consists of about 10,000 sarcomeres, and each sarcomere consists of precisely arranged myofilaments. When viewed under the light microscope, skeletal muscle has a banded or striated appearance. This is a result of the arrangement of the myofilaments contained in each myofibril. The dark bands of striated muscle are called A bands, and the light bands are I bands. The A and I designations have to do with the way the proteins in the bands bend light. The I bands contain only thin myofilaments and have a Z line or Z disc running right through their middle. Each sarcomere extends from one Z line to the next. In other words, a sarcomere is the region of a myofibril between two successive Z lines. Each band contains a region of overlap between thin and thick filaments and has a light region in its midline called the H zone, H for hell, which means bright. The H zone contains thick filaments only. Looking more closely at the molecular makeup of the sarcomere and the I and A bands, we can see that multiple proteins are involved. The Z line is composed of discs of a protein called alpha-actinin. It is responsible for anchoring the thin filaments, which are mainly made of actin. Another filament found in the I band is an elastic filament composed of a huge protein called titin. The titin extends from Z disc to the thick filament and attaches to the M line, which consists of proteins that link the thick filaments together. The A band therefore consists mainly of thick filaments, the M line, H zone, and a zone of overlap between thick and thin filaments. This slide shows the detailed structure of one sarcomere in longitudinal view D, and cross-sections through the different zones and bands showing the relationship of the various types of filaments to one another. In addition to being multinucleated, numerous mitochondria are located between the myofibrils. These provide the energy to power muscle contraction. There is also an extensive smooth endoplasmic reticulum which forms a meshwork of tubes surrounding each myofibril. Compared to the smooth endoplasmic reticulum found in more typical cells, the sarcoplasmic reticulum of muscle tissue has a unique function. First, the sarcoplasmic reticulum sequesters calcium ions. This means its membranes are loaded with transporters which have the ability to pump calcium ions into the sarcoplasmic reticulum against steep concentration gradients. The total concentration of Ca2+ inside the cisternae spaces of the sarcoplasmic reticulum can be 40,000 x more than that of the sarcoplasm. The calcium is stored in the SR and is released on demand when the muscle fiber is stimulated to contract. Calcium is the final go signal for muscle contraction. Most of the tubes of the SR run longitudinally along the myofibril. Others form larger, perpendicular cross-channels at the A-band-I band junctions. These enlarged channels are called the terminal cisternae, end sacs. The sarcoplasmic reticulum is connected to the sarcolemma by extensions of the sarcolemma called transverse tubules. We will see that the transverse tubules conduct the signal to contract, or action potential, from the surface to the interior of the muscle cell. Transverse tubules, or T-tubules, are unique to muscle tissue. The lumen of the T-tubule is continuous with the extracellular space. Each T-tubule runs into the cell between the paired terminal cisternae of the SR-forming triads. Thus, triads consist of three membranous structures: terminal cisterna, T-tubule, terminal cisterna. So, how do our muscles contract? We take for granted being able to walk, dance, play sports, grasp objects, or pick up a cup of Starbucks, but these activities have their beginnings at the molecular level. We can apply what we know about how skeletal muscles are put together, that is, the structural levels of skeletal muscle, to shed light on our current understanding of muscle contraction. As the thin filaments slide over the thick filament, each sarcomere gets shorter, each myofibril gets shorter, then each muscle fiber gets shorter, causing the fibers in a fascicle to get shorter, and finally, the entire muscle shortens. In our next presentation, we will learn how this happens.